I know, it hates us.
No, it doesn't. We're live.
Oh, yes.
Sweet. I'll start. So, welcome. This is Wesley and Clark. We're going through Group 5 lesson problems. So, for number 1, this is our work. So, the actual problem says, one second. That one's having a struggle. That's what the actual problem says. So our number A says determine the allele arrangement for the heterozygous parents, and so pretty much you just find the one that has the most offspring. And so for us, that was the agouti and mahogany and wild type for one parent, and then the wild type, ragged, and wild type for the other parent. For letter B, it says determine the gene order, and pretty much you just do that little cross and determine which one of those 3 will line up with the least expected allele order. And we found it to be the A in the middle, so it would be the MARW or the RWAM, either way. And then the other one was the MWAWR. For C, you do the gene mapping. So you find the distance between the M and the A, the mahogany and the agouti gene or allele. You would cross the M side. And so we got the ragged, agouti, we got the mahogany, wild type, and ragged, and there's 12 of those. And then we got the wild type, agouti, and wild type, and there's 13 of those. And you use that little equation, so it's the 2 recessives plus those 2 that you got from the crossover, all added together over the total number of offspring, which is 206. Times 100, you get 13.1. When you cross over between the A and the R, we got 16 and 18. And after the equation, we got 17.5. So between the R and the A was 17.5 MU. And between the A and the M was 13.1 MU. And then for D, put both of those answers over 100, times it by the total number, and then do 1 minus 2 over that answer, and we get 57.6%. That's number 1.
Number 2 is literally the exact same. But we're talking about these Drosophila fruit flies again. And so we've got the SC for scoop bristles, the S for sable bodies, and the V for vermilion eyes. And then all the other wild type versions of those. So you just do the exact same thing. So you just find the ones that have the most. Right here. And so that's our answer for A. The 314 one has the SC plus, the S and the V, and then the 280 has the SC, the S plus, and the V plus. Then for B, you do the little squiggly dip things. And then you find the 2 that are the double crossovers, and they're the ones that have the least. So we got the 10 one and the 14 one right there. So the 14 one had SC plus, S plus, V plus, and the 10 one had SC, SV. And so that told us to choose the one where the SC was in the middle for our order. For V, we took that, did the crossovers, and found the number of each that we had and put it into that equation right there. This is the map that we drew out. So we knew that the distances between the V and the SC was 10 map units, and the distance between the SC and the S was 33 map units. And then for D, we then took those answers from C, put them into our equations, solved it all out, found that I was 27%. And that's question 2.
Number 3 actually was a little bit tricky. Pretty much the same question, except for there were a couple of offspring that they didn't get any, like a couple of genotypes that they didn't get any offspring for, or phenotypes, sorry. So originally we were working through it and we got to the crossovers, and on one of the crossovers, it was just wasn't there. So like, wait a minute. So we had to go back and look. And I guess when there's just a glassy eye or a coral body and striped thorax, those 2 phenotypes didn't have any progeny or any people that had that. So with that problem, we had to use those 2 zeros that weren't even listed on there. So there's not any offspring for them, so there's zeros, and that helped. We had to go back and redo B. So eventually this is what our B looks like. I had to go up through there, and you can see we found that the S? Yeah, the S in the middle was for the gene order. For C, we did the mapping according to that, and for D, we used the similar equations we've been using. So that one was interesting because the interference value was actually 100%, which is kind of different from all the others. All the others were like 50 or 60%. And that was number 3.
And then number 4 was the same, but back to not being quite so tricky. So then again, we took the ones that had the most, found their phenotype things. So for the 385, we got that it was anther, and then wild type, and then for the 379, we got that it was wild type, and then brachytic and fine. Then we did our little whatever looping, swapping, crossing overs, and took the ones with. We're back. I don't know if you saw that, but we took our little loopy dip things. Stop laughing. We took the double crossover ones. So for one of those, there were only 2. That was the wild type, brachytic wild type, and then there was one with 3 that was anther, wild type, and fine. So then we found the one that swapped them right, and that's the one that has F in the middle. So your gene order should look like this, or the reverse of that. It doesn't matter as long as the F is in the middle. And then C, we took all the crossovers, added up their values, divided them by the total number, times it by 100, got these numbers, and then drew our gene map, or our map here that showed a distance of from the BR to the F. That was a distance of 5.1, and a distance from the F to the AN. That was a distance of 19. Plugged all of that into our equations for D, and found out that our interference value was 48.5%, or 48%, somewhere in there, depending how you round.
For number 5, it's a bunch of interesting questions. This is all of our work for 5. So for A, it gives you a genotype, the big A, little a, big B, little b. Says they're all those. The A's are on a homologous chromosome and the B's are on a separate homologous chromosome. Says what's the possibility of each of these gametes in one cell after meiosis, pretty much. So if crossing over occurs, there's essentially a 25% chance that any of those could occur. So that's where we showed this crossing over right here. And to make it sense in my mind, I just did a Punnett square, and it was just all 25% for all of those different genotypes. For B, it says, suppose it happens, but crossing over does not occur. So really, it would just have those 2 genotypes because the hybrids wouldn't be able to cross over. For C, it says
they're linked, the crossing over does occur.
It says they're linked. Crossing over still occurs. And on C, we determined that it would be, I guess all four could still be produced even though they're linked because crossing over still occurs. For D, it says that the A to the B is 7.8 map units apart, so that means there's a 7.8 chance that crossing over will occur on those. And so for those hybrid ones, the uppercase and lowercase combinations of A and B, pretty much the big A with a little b or the little a with a big B, there's 7.8 chance that both of those could occur. So for one of those to occur would be half of 7.8, so we got 3.9 on that one. And then for the big A, big B, and little a, little b, those there's a 92% chance that those both will occur. So for one of those to occur, the big A, big B, it's half of 92.1, so it's 46.1%. And same for the other one. That's our group problems.